Hi everyone. On the last video, I didn't get a chance to really talk to you about the structure of DNA once it has formed its strands into the double helix. And that in itself conserves a lot of area for that molecule, but it's not nearly enough area for the conservation of DNA packed into the nucleus. As we mentioned, the strand of DNA that you have in your nucleus is roughly 100,000 times the size of the nucleus. So you can see that there's an enormous amount of geometry involved in getting this to package into that such a small area. So after we have used that double helix structure, then that double helix fiber will wrap itself around proteins that are already produced. And in wrapping itself around proteins, it creates a complex called chromatin. So the proteins are called histones. The DNA wraps around it. The complex is called chromatin. And that will fall back on each other, and that will turn around each other and form into beads that wrap around. And those beads that you see right here, in here, are nucleosomes. And all of that wrapping produces a coil that produces a fiber in here. And that fiber loops around a scaffolding to form a chromatid. 2 chromatids on a chromosome. So you begin to understand that the double helix is just one part of the geometry. The looping around the proteins is another huge space saver, and then the coiling in the nucleosomes is another form of it. I hope that helps you out. That is in your background information, and it will help you in your report sheet. To get into the structure of DNA and the questions for the report sheet, we have to get into the procedure now. So there's 7 parts to the procedure. And the first is you have to leave enough time to chill your fruit and chill the ethanol well. And there's nothing about chilling the fruit in the procedure, but I strongly advise you to chill the fruit. It will work better. Just put it in the refrigerator. Don't put it in the freezer. Freezing just destroys cells and it won't help you. So just chill it in the refrigerator for an hour or so. Make sure that the ethanol is very cold. That can go in the freezer. Then you're going to prep your buffer. And the third thing is you're going to break down the cell walls because you're using a plant here. Then you're going to lyse or slice into the cell membranes, plural, I should change that there. And you're going to then filter out the larger fragments of the cell and purify and precipitate the DNA and then harvest and photograph the DNA. To do all this, from your kit, you're going to need your small graduated cylinder. You're also going to need your big graduated cylinder, your 100 cc graduated cylinder, your 250 milliliter beaker, all the materials in your bag. I would suggest using deionized or filtered water for your water that you need. And I think that's it for that. Very important, you are going to need your safety goggles and your gloves. Right? You don't want to get ethanol on your hands. It'll just dry them out so much. And you don't want to get ethanol on your skin if it ever splashes, if somebody bumps into you. You also don't want to get it on your clothes. So wear your bib and wear your safety goggles. Okay, very important. Ethanol is flammable. You do not want to do this near your stove. Do it on an island, do it at a table. Do not do this experiment near a stove. Ethanol is not regular alcohol, so don't drink it afterwards. You'll wind up in the emergency room. I'm an EMT, happens. You don't want to get your stomach pumped, so don't drink ethanol. This is extremely high percentage of ethanol. You don't want to do that. Stomach pumping is really not a lot of fun. Like I said, you have to chill the ethanol and you have to chill the fruit as well. Right? Hold on a second. Okay. For our first part of this, we have a lot of drilling to do. We have to get through at least 4 walls. Well, some of the walls are thicker than others, but DNA is very well protected within the cell for good reason. And we don't want viruses to get into the DNA and disrupt it. We don't want wavelengths of light to get into the DNA and disrupt it. So it is very, very well protected. The first layer of protection for plant cells is on the outside, and it is the cell wall, which is heavily fortified. And sometimes there are 2 cell walls, which are heavily fortified with fibers of cellulose. Very difficult to break. And then right inside of the cell wall or cell walls, there is a cell membrane, which is identical to the animal prokaryotic cells. The animal eukaryotic cells. Also has a cell membrane. And then you have what's called a nuclear envelope, which is also called the nuclear membrane. Same thing. And there is a membrane protecting the DNA within the nucleus. So you have a lot to go through. There are 1 to 2 cell walls, 1 cell membrane. There are 2 nuclear membranes. And let's go into that a little bit more. The cell wall is tough to break, made up of cellulose fibers, a very strong carbon bonding relationship there. And that's on the outside. These crisscross over to add even more strength to that wall. Inside, the plasma membranes is the umbrella term for cellular membranes and nuclear membranes, and cellular membranes around organelles inside the cell. So plasma membranes is a catch-all umbrella term for whatever membranes are inside the cell. They have in common a phospholipid structure. The phosphorus head, the hydrophilic head, the negatively charged phosphate head is generally shown as a circle in most biological diagrams. The hydrophobic tail, the lipid tail, is generally shown as legs, sometimes with a kink in it. These line up in a layer, too deep, and sometimes you can have 2 membranes next to each other, particularly around the nuclear membrane. And these hydrophobic tails line up together to keep out water. And the hydrophilic heads are on the outside because they're usually impacted by saline. This is the setup for the cellular membranes and the nuclear membranes. Very similar all the way around. You need to break through the cell wall, maybe 2, and the cell membrane and the nuclear membranes. Got a lot of work to do in this lab. To look at this lab, the second thing we need to consider is that we need a buffer solution to try to help us to break through the cell membranes. And you need to make this up before you start the lab. So you'll need your 25 ml graduated cylinder. You'll also need your 100 ml graduated cylinder. You'll need your filtered or deionized water if you can get it. If you cannot get that kind of water, please let your water, you'll need about 200 cc's. Please let that sit out overnight. so that the minerals from the water tend to precipitate out. And you'll probably see them on the bottom. But please do yourself a favor. You'll save time in the long run if you precipitate that water out overnight if you can't filter it or deionized water. You'll need your detergent. You'll need your salt package as well. You'll make it up according to these instructions and set it aside. The reason you are doing this, you're making a buffer, and a buffer is a chemical that can easily release hydrogen ions into a liquid or take up hydrogen ions in a liquid, or both. Some buffers are very versatile and can do both. In this particular solution, salt is the buffer. Salt can do both. And the reason we are using a buffer is because we are trying to maintain a physiological pH, right around 7.4. So we want to resist big changes as we do our chemical reactions, and our salt helps us to do that. The other ingredients in your buffer, and this goes right back to your report sheet, are a detergent, and it's not just any detergent, okay? This detergent happens to have, well, and most of them will, but this one happens to have sodium dodecyl sulfonate, and that is specifically known for breaking hydrophobic bonds. So, for instance, when you hear that Dawn detergent is good for cleaning ducts on oil spills, it is, because it has a very high percentage of the sodium dodecyl sulfonate. Other detergents may not have as high as a percentage. This has a pretty low percentage, which is good for us. And that breaks up grease on your dishes. Well, guess what? It means it is breaking up fat layers. And if it is breaking up fat layers, it is breaking up the fat layers in your phospholipid cellular membrane, and you can get to your DNA a little easier. That's why we're using the detergent. The water is polar for solubility of the salt and of the detergent, and it's the universal solvent. So one of the questions in your report sheet is dealing with that. And I'll see you on the other side.